The effects of microplastic pollution in water quality and aquatic ecosystems of Laguna de Bay, Philippines.
Is this fish really that healthy? What are those tiny little things in its stomach? That's plastic, microplastic. But how did it get there? 8 million tons of plastic litter enters the ocean annually. This plastic eventually breaks down into microplastic. What are microplastics? Microplastics are really tiny pieces of plastic with a size range below 5 millimeters. Microplastics are ingested by zooplankton and other small sea creatures. This is how they enter the food chain. Microbeads or microplastics are used in shampoo, soaps, toothpaste, and face wash. They make their way through drains and rivers into the ocean. Wind, water pressure, and UV rays break down plastic into microplastic. Even car tires shed plastic dust. Microplastics are so small and non-biodegradable, it's everywhere.
Laguna de Bay, the largest freshwater lake in the Philippines, is located southeast of Metro Manila, the capital city of the Philippines, with an area of 911.7 square kilometers. It is economically important body of water that supports approximately 9,000 hectares of fish span and provides a source of income for fishers in Laguna and Rizal provinces. Somewhat shaped like W, the lake is split into four bays: the West Bay, Central Bay, East Bay, and South Bay. Talim Island separates the West and Central Bays. This division are caused by significant bathymetric differences between these areas. Moreover, Laguna de Bay serves as a source of irrigation water, industrial cooling water, hydroelectric power generation, transportation route, recreational venue, a source of animal feeds and fish supply, and a source of domestic water supply for the neighboring provinces, including the capital city, Metro Manila. However, the lake's importance has placed it in peril from a host of problems, including pollution from untreated sewage and industrial waste, overfishing and sedimentation, and microplastic pollution.
Microplastics enter the lake through sources like industrial effluents, urban runoff, and improper waste disposal, traveling via river systems, stormwater, and biological uptake. Knowing the sources and pathways of microplastic pollution is crucial because it helps identify where the pollution originates and how it travels through the environment. This understanding is essential for developing targeted strategies to prevent further contamination, protect aquatic ecosystems, and safeguard the health and livelihoods of communities that depend on Laguna de Bay. It also provides valuable insights for policymakers to implement effective waste management, pollution control, and environmental protection measures. The first example of the sources of microplastics in Laguna de Bay is the industrial discharge. Industrial discharge around Laguna de Bay releases microplastics through effluent discharge, particularly from textile, plastic production, and manufacturing sectors. Second is the urban runoff. Microplastic from urban runoff, including particles from degraded plastic products, synthetic textiles, and road debris, enter the lake via storm drains. Third is the wastewater treatment plants, where wastewater treatment plants may not fully capture all microplastics, leading to direct discharge into the lake through treated effluents. Next is the agricultural runoff, where microplastics from plastic waste used in agriculture, like plastic mulch, irrigation, tubing, can enter the lake during heavy rains or flooding. Next is tourism and fishing activities. Improper disposal of plastic waste by tourists and fishing activities, including plastic nets and gear, contributes microplastics to the lake. Last sources of microplastics in Laguna de Bay is the domestic waste. Household plastic waste, such as packaging and synthetic fibers from washing machines, may end up in the lake through improper waste disposal. Moving on, the pathways of microplastic pollution refer to the routes through which microplastics travel from their sources to Laguna de Bay. Understanding these pathways is critical in assessing how microplastics are transported and accumulated in the lake, impacting water quality, aquatic organisms, and the overall ecosystems. The first pathway of microplastic in Laguna de Bay is the river system. Rivers, especially the Pasig River, transport microplastics from industrial, urban, and agricultural sources into the Laguna de Bay. Second is the stormwater runoff. Microplastics from urban streets and commercial areas are carried by rainwater through stormwater drains and into the lake. Third is the direct dumping and shoreline pollution. Improper disposal of plastic waste along the shoreline and surrounding areas contributes microplastics to the water. Next is the sedimentation, where larger microplastic particles may settle at the bottom of the lake, potentially affecting the lake's sediment and aquatic life. Fifth is the biological uptake. Microplastics are ingested by aquatic organisms, including fish, shellfish, and plankton, which can bioaccumulate these particles. Last is the water currents. Water currents within the lake can disperse microplastics across different areas, influencing their contribution or distribution and concentration.
The first documented evidence of microplastic in the largest lake in the Philippines were found out through the study of Argadio et al. In the year 2022, entitled Microplastics in the Surface Water of Laguna de Bay. They found out that all the microplastics were pieces of plastic used by the most of the community, with the major polymers being polypropylene, ethylene vinyl acetate copolymer, and polyethylene terephthalate. A majority of the plastics were small colored particles, specifically blue-colored microplastics, which pose a threat to water quality and fisheries of the lake, as this can easily enter the food chain. So, what steps were taken in that study? First, they used a plankton net to collect microplastics on the surface water of Laguna de Bay, following the methods of Persec et al. In the year 2016 with some modification. A plankton net with a mesh size of 20 microns was used to each sampling site. The boat traveled 10 meters while the net was set at a depth of 20 centimeters to keep the entire net submerged in water. GPS coordinates were also recorded at the start and end to aid in calculating the distance traveled. Sampling was carried out in 10 sites, shown in this figure. Sampling stations were grouped according to the lake's bathymetric differences. The four groups were South Bay, West Bay, Central Bay, and East Bay. South Bay was composed of Station 1 and Station 7. West Bay was composed of Station 2, Station 3, and Station 4. Central Bay was composed of Station 5 and Station 6. And East Bay was composed of Station 8, Station 9, and Station 10. Within each sampling site, water samples were collected, yielding 10 samples for microplastic analysis. For the analysis, in the laboratory performed, all glassware was washed thoroughly with distilled water for decontamination. Solids collected in the plankton nets were soaked in potassium hydroxide solution and heated in a 60 degrees Celsius oven for 24 hours to digest organic material found during the water sampling.
The sample is then floated to extract the plastic debris using density separation and sodium chloride at a 30% concentration. Vacuum filtration using millipercent was performed after flotation. The probable microplastic particles were collected into a clean Wattmann glass filter. Each glass filter was washed with distilled water and oven dried until dry. A clean Petri dish kept the filter paper for optical microscopy analysis using 40 times magnification. The particles' shape, size, and colors were also measured and identified. Additionally, this figure shows the types of microplastic assessed in surface water of Laguna de Bay. A are called filament, B are fragments, C are fibers, D are granules, and E are films. A film was characterized as a tiny, very thin coating or a substantial chunk of plastic trash, while a fiber was described as microplastic with a long, slender appearance. Pellets were described as spherical, rounded microplastic particles. The classification of fragment was used when a microplastic could not be classified as a fiber, pellet, or film. This technique was based on the classification method used by Su et al. In the 2016. All laboratory activities were done at the Chemistry Department of Caraga State University, Butuan City, Philippines.
Microplastic pollution has become a growing concern, not only for the environment but also for human health. These tiny plastic particles, less than 5 millimeters in size, can enter the human body through various exposure routes such as ingestion, inhalation, and dermal contact. Once inside the body, microplastics can accumulate in tissues and organs, potentially causing harmful effects. They can carry toxic chemicals, heavy metals, and even harmful microbes, leading to issues such as inflammation, oxidative stress, and possible DNA damage. In this presentation, we will explore how microplastics impact human health and why understanding this risk is essential for safeguarding public well-being. This infographic highlights the adverse impacts of microplastics on human health by acting as carriers of harmful substances such as heavy metals, antibiotic resistant microbes, and toxins. It emphasizes the various pathways through which microplastics can damage the body. So we have the antibiotic resistant microbes, where microplastic can carry antibiotic resistant bacteria, contributing to the spread of antimicrobial resistance or the AMR, which makes infections harder to treat. We also have the neurotoxicity, where microplastics can potentially damage the nervous system, leading to neurotoxicity, which affects brain function, causes issues like memory loss, cognitive decline, and neurological disorders. We also have the microbial toxicity, where microplastics can introduce toxic microbes or increase harmful bacterial activity, causing microbial imbalances in the gut and other organs. Next is the metabolic syndrome. It is the exposure to microplastics may contribute to metabolic syndrome. It is a group of conditions that increase the risk of heart disease, diabetes, and obesity. Genotoxicity is the microplastics can damage genetic material DNA, causing genotoxicity, which may lead to mutations, cancer development, or reproductive issues. We also have the uterine arterioles reduction. In females, microplastics may affect the uterine arterioles, possibly leading to reduced blood flow to the uterus, affecting fertility or reproductive health. Inflammation. Microplastic induce inflammatory responses in various tissues, leading to chronic inflammation, which is associated with a wide range of diseases. We also have the oxidative stress, where microplastic can increase the production of reactive oxygen species or the ROS, leading to oxidative stress, which damages cells, proteins, and DNA. Endothelial dysfunction. Endothelial cells line blood vessels, and exposure to microplastics may impair their function, contributing to cardiovascular disease like hypertension. Last is a chronic disease. The accumulation of microplastics can trigger the development of chronic diseases, including cardiovascular disease, diabetes, and cancers. This diagram illustrates how microplastics pose serious risk to human health by affecting multiple systems including the nervous, immune, cardiovascular, reproductive, and metabolic systems. They cause damage through mechanisms like inflammation, oxidative stress, DNA damage, and disruption of microbial balance. These effects underscore the need for reducing exposure to microplastics to prevent long-term health complications.
We can do something.
We can reverse this damage. Reduce, recycle, rethink. It's the small changes that create a big difference.